Okay, great. Well, good afternoon, everybody. It is, depending where you are, 12:45 p.m. We have an amazing lineup of speakers today and not a lot of time. So I'll just jump right into it. My name is Georgia Peck. I will be chairing today's session, and I am the Lakes Program Director with Living Lakes Canada. We are based in British Columbia, which is where I'm calling from today, specifically Armstrong, BC, which is kind of the northern end of the Okanagan for anybody who doesn't know. Like I said, we have a pretty short amount of time today, so I'll be presenting or introducing the presenters before they get going. I'll be timing you as well, and in the chat box, I'll give you a 1-minute warning before you hit that 10-minute mark. And then we'll move into the lightning talks afterwards. I think we'll leave it till the very end for questions or discussion if there's time, just so everybody can get through their presentations. But I do encourage presenters to include their contact information into the chat box. Then any questions or discussion points can be directed specifically to the presenter that you would like to discuss with. So with that in mind, we can move forward with our first presenter, who doesn't look like she's on the call yet, Tia Jenkins.
Yes, I'm here on behalf of Tia.
Okay, perfect.
Yeah.
Take it away, Stephanie.
Okay, thank you. I'm just gonna have to remember how to share my screen. Here we go. It's been a while. Okay. So yeah, I'll try to stay in time here. So today I'm just going to be sharing some work. Tia couldn't make it today, and there's a team of people that were part of this work, and we're obviously funded by Global Water Futures, as well as largely by a microplastics fingerprinting project at the University of Waterloo. And I'm in the Grand River watershed right now at the University of Waterloo. So the question driving our research was, where are the microplastics data? With asking this question, the goal of asking this question was that microplastics data can support water quality management and environmental policy. And so, as I mentioned already, that question is relevant, very relevant to developing good policies. And microplastics is an emerging field and rapidly growing. You can see the number of publications. You've probably seen this before. And the exponential increase in those publications. So while there's a large amount of research being published, as I said already, that question is, are those data there to support the research and future research? So are those data that are being generated findable and accessible? So this example is from the USGS, where they show that kind of data where records are missing, and they give this example for nutrient water quality data. There's a cost of billions of dollars worth of data that are no longer usable, even though all that effort went into collecting them. So we're kind of inspired by the FAIR data principles in our analysis, and the FAIR data principles, I'll outline them really quick, that they ask, are the data findable, accessible, interoperable, and reusable? And our focus was really on the first two pieces of FAIR. We weren't able to assess the interoperability or the reusability of the data. But these standards are for describing, or these FAIR principles are about describing best practices for data description, interoperability, and how it can be reused by others. And so the question that we then asked with these FAIR principles in mind were, to what extent are the data that underpin environmental microplastics research articles openly shared? And so to what extent are they therefore findable and accessible? To assess this, we looked at articles published about microplastics using the search term microplastics kind of in Web of Science. And we randomly selected 20% of around 7,000 peer-reviewed articles. And with these 20% of articles, we assessed, is there a data sharing statement included? And if yes, how are the data being shared? And then we also looked at microplastics data sets in data repositories again, we searched a generic microplastic search term. We looked in 10 different repositories. The list of them is here. And again, we were assessing how accessible those data were. So the data sharing trends in the peer-reviewed articles look like this, where there were only in total 8 data sharing statements between 2006 and 2012, but that's also because there weren't very many articles being published at that time. From 2013 onwards, the data sharing statements began to increase, and also the proportion of the data sharing statements in the total articles being published started to increase. And then again, from 2019 onwards, we see that there were about half of the articles that had a data availability statement included in them. So when there are data availability statements included in an article, how are the data being shared? What is described in the data availability statement was the next question we asked. And you can see here that 38% of the data availability statements said that the data were available in the supplementary files. Next, most available data were in the article itself. Then you had to request the author, and only 13% were available in a repository, which is one of the better ways to share data. Requesting via the author is less ideal in terms of openly available data. And same is true for the supplementary file data availability statements. What we saw when we looked at the publications available in the repositories versus, or the data in the repositories, which weren't always linked to publications clearly, that's also an issue. We saw that they didn't increase at the same rate as the rate of increase in publications. So this is an issue because, sharing data in repositories is the best way for sharing data generally, because repositories have really good data sharing standards. And then we kind of asked the question, but we weren't able to answer it. This is a big question. Are open data being adequately described? So this is just to show you the distribution of where the microplastics data are being collected that we identified, and the distribution of media types. So you have sediment, you have water, but you also have animal tissues like fish and mammals and birds. So there's all sorts of media types and therefore data types and therefore metadata attributes that are needed. So it's kind of a grand challenge. Kind of overall, to wrap up, we showed evidence that data sharing practices are increasing, but we identified that there are challenges that will need to be addressed to ensure the long-term kind of availability or findability and accessibility of data. So some examples of things that will need to be addressed: there will need to be standard vocabularies for kind of data types and metadata attributes. And this can also feed into having standardized metadata / data reporting templates. And so, of course, fair data will be a vital baseline for future monitoring and decision making. And we think that Global Water Futures is well poised to support the growing data management culture in Canada and to help the microplastics community address these challenges. We know, obviously, the tri-agency research data management policy is now calling for open data sharing. So microplastics is not alone in needing to face this challenge, but we wanted to highlight those gaps for environmental microplastics data specifically. And that's everything. So thanks.
Wonderful. Thanks so much, Stephanie. And like I said, we'll wait till the end for any questions or discussions. Stephanie, if you are comfortable putting your contact information into the chat box in case we don't have time for questions, please feel free to do so. If you want to end sharing your screen, I'll pass it on to Nan Zhang, who can share hers if she has a presentation.
No questions? Okay, so I will get started my presentation, and it's a pleasure to share my jobs about the production of the high quality drinking water using the membrane technologies. And this project is funded by the Global Water Futures and supervised by Prof. Illinois at McMaster. The membrane has been used as a filter to produce the drinking water since the last century. It has the advantages of the small footprints and the simple modularity at the lower operational costs. However, the membrane fouling is a big challenge for the long-lasting operation. We can find out certain particles such as the bacteria, proteins, and the scaling ions was retained on the membrane surface, reducing the membrane permeability and increasing the energy consumption. How can we resolve this challenge? We can directly change the flow directions by using the mechanical forces to remove the foulants on the membrane surface. In comparison, new types of the membrane, the electrically conductive membranes, are well developed for the fouling mitigation. The electrostatic repulsion determines the effectiveness of the cleaning strategies, and the in situ fouling control can be achieved. And different types of the conductive membranes were developed using different conductive materials, such as the carbon nanotube and the metal films. In the following, I will introduce the water purification process using the membrane technology and the electrochemical techniques. So the first step is to fabricate the carbon nanotube membranes. So this membrane is fabricated by dispersing the carbon nanotube into water, and this suspension was filtered through the commercial membranes. So the CNT layers will develop on the top surface. Via this pressure-driven deposition, the CNT membranes showed the higher surface conductivity. Another types of the conductive membranes is the gold-coated membrane. So this ultra-thin metal film was produced on the porous membrane substrates via the sputter deposition, and the gold-coated membrane exhibits the desirable surface conductivity and permeability. And compared to the CNT membranes, this ultra-thin membrane have the 10-fold higher conductivity and 12-fold membrane permeability. So next part is the introduction of the electrochemical techniques in order to sense the membrane fouling evolution on the membrane surface. So this technique is the electrically impedance spectroscopy. This EIS is a non-invasive technique in order to sense the fouling development. We can regard this EIS as a black box. So a very small amplitude alternating current potential was injected to these electrochemical systems, and the resulting current can be achieved as the output signals. The differences between the input and output signals, such as the phase angle and the different amplitude, can provide us the interfacial properties on the membrane surface where the fouling occurs. Further, we can using the equivalent circuit to interpret this impedance data. So that means the fouling phenomena on the membrane surface and in the concentration polarization layers can get well interpretation. So our target is to establish a membrane system for the fouling sensing and fouling mitigation simultaneously. So this is our whole target. So the experiments are as follows. Because the biofilm was regarded as the most stickiest foulant on the membrane surface, so we're using the bacteria to challenge the membrane filtration process. The bacterial culture harvested from the tap water was used as the feed and then pumped into the membrane module, where the conductive membranes and the working electrode and the graphite and the counter electrode. The biofouling experiments was carried out under a constant transmembrane pressure over a duration of 12 hours. And we recorded the membrane flux and the impedance data. When the biofouling was accumulated on the membrane surface over time, the permeate flux was reduced. And correspondingly, for the impedance data, the impedance in the low frequency region reduced. And then we're using the circuit to interpret this impedance data. And we found that the diffusion-related impedance can be sensitively monitor the fouling development on the membrane surface. And then we compared the changes of the normalized impedance and the normalized permeate flux. The impedance-based detection is more sensitive than the flux-based detection, especially in the initial stages of the fouling evolution. The next part is we try to apply the cathodic potential on the membrane surface to repel the biofoulants on the membrane surface. And we found out after applying the intermittent potentials, the normalized flux was recovered. And so we can get the conclusion that we successfully established a two-electrode membrane system, and we confirmed the abilities of conductive membrane as the dual platform for the fouling sensing and the fouling mitigation. And the fouling sensing was achieved by using the electrochemical techniques, and the fouling mitigation was achieved by applying cathodic potentials. So this conductive membranes is very promising for the next generation water purification process. So last but not least, we accept the support from the Illinois lab and the funding from the Global Water Futures and the technical helps from the CCEM Center at McMaster. Thank you.
Great, thank you so much, Nan. That was fantastic. Like I said as well, for anybody who's joined since the first introduction, we'll wait until the end of all presentations, including the lightning talks, just to ensure everybody has time to present. And again, Nan, if you're willing to put your contact information into the chat box so people can direct questions specifically to you, please feel free to do that. So next up, we have Heidi Swanson.
Excellent. Thank you so much, Georgia. And can you just confirm that you can hear me and see the screen?
Yep, everything looks and sounds great.
Awesome. Thank you. I will just give folks a bit of a warning in that the internet is a bit unstable where I am. So if I peace out, I'll peace back in within about 30 seconds. So thank you so much for having me here in this session. I am just the face of a large project, and a lot of these folks have really led a lot of the work. So collaboratively with several First Nations within the Detroit First Nations in the Northwest Territories, and a lot of collaborators, co-authors, and students. And I would especially like to send some thoughts to some of the folks on this list who have endured catastrophic flooding in Hay River this week and are currently out of their homes. So sending our best thoughts of resilience to them. I want to talk a little bit about a little side project that has emerged after about 10 years working in the Dachau region of the Northwest Territories on questions around fish mercury. So concentrations of mercury in fish are a concern because fish are a staple dietary food source, and exposure to fish-derived mercury can pose human health concerns at high enough levels. And so in the communities where we're working, folks want to know why some lakes have high fish mercury and other lakes have low fish mercury, and then how climate change and resource development is going to affect those mercury levels. And in lakes with high mercury, is there anything we can do about it? That's kind of the 30,000-foot overarching view of the project. And I'm actually going to really zero in on a reductionist question, but that could have far-reaching implications for perhaps our next 5-10 years of work. So what we do in this project is we began in 2012 and we build off foundational community questions centered around these overarching kind of objectives. And we've managed to sample 13 lakes between 2013 and 2021 together with Indigenous guardians. So the lakes represent 4 ecoregions, which means we've been able to capture quite a bit of environmental heterogeneity, both in water chemistry and in catchment composition. What happens at each camp is that the university crew goes in. We're in the southern region of the Dachau here. A university crew goes in and we work for 2 to 3 weeks on the land each summer to sample about 2 lakes with Indigenous guardians from communities for which the lakes that we're sampling that year are part of their traditional territory and that they harvest for subsistence foods. It's pretty involved. So at each lake we go to, we sample fish, lake water, mercury and chemistry, sediment, benthic invertebrates, and then we get back to the ranch. We characterize the catchments and look at lake morphometry. And then we try to figure out, mostly by Mehdi, a student that I'm going to plug who's giving a talk tomorrow, how all of these factors interact to affect fish mercury concentrations. So yeah, these camps, sorry, I'm also supposed to be in a Teams meeting. These camps are on the land and in the bush. So we usually fly in, build a camp. Some of them we can quad or drive to. And then because we're on the land for several weeks at a time, we can actually start to answer some of these opportunistic questions or just kind of take samples out of curiosity, which actually has resulted in some interesting things. So again, a little bit of a spoiler for Mehdi's work, but I'm going to plug him a little bit here. So what's going on here is that we have figured out through these 10 years of data via Mehdi that catchment factors interact with lake factors, which then drive 2 main variables. And these 2 main variables explain more than 80% of the variability in fish mercury concentrations. But what I'm going to talk about here was this very curious result where we found that sediment mercury wasn't that well, or in some cases at all well or not, related to water. So methyl mercury in water. Why was this confusing? We think that sediment and water mercury should be really closely related because the conventional understanding of mercury cycling in lakes is that you have mercury in the atmosphere in the form of gaseous elemental, or you've got mercury 2+, and that can fall directly onto the lake surface or onto the catchments. And then that mercury 2+, whether it's from the catchment or the atmosphere, goes to the bottom of the lake, and this is where it's methylated. Because inorganic mercury is not toxic and it doesn't bioaccumulate or biomagnify, it has to be methylated first. And we thought that this happened in anoxic bottom water sediments where, sulfate reducers, ion reducers, and methanogens would add this methyl group. Further, we thought that most of the DOM and most of the methyl mercury was sourced from within the lake, so autochthonous. Now, this was thought to be kind of the prevailing paradigm until the last 5, 6, 7 years. When, of course, as usual, we discovered that this conventional understanding was based on lakes that are within a few hours of universities and research stations in temperate areas. So when we start broadening out to consider lakes that more fully capture the diversity on the landscape, for these like shallow, unstratified, catchment-dominated lakes, methylation actually occurs in the littoral sediments and in littoral macrophytes and benthic algae rather than in these profundal sediments. And we had sampled here in the lakes at the center, because this is where we thought we would find the relationships. But in fact, it's a lot more nuanced in these subarctic lakes, we think. And also it looks like DOM in these lakes and methyl mercury is much more allochthonous source rather than a toxinous source. So this is from a paper published by Branford et al. in Stoughton in 2020. And basically what they did is they added an endpoint to our understanding of how mercury cycles. So we thought, well, we've got this 10 years of data. We didn't really collect it for this purpose, but let's see if any of this starts to hold true. And if so, we can develop a new hypothetical framework for going forward. So we thought, okay, if primary sites of in-lake methylation are littoral rather than profundal, then our benthic invertebrates should have higher methylmercury in littoral habitats. And when we look at percent methylmercury, which is a proxy for net methylmercury production, we did in fact find that benthic invertebrates had significantly higher mercury in these nearshore littoral environments where we thought methylation wasn't really happening, but looks like it is. This holds true when we control for functional feeding group and taxonomy as well. Our second prediction then was if catchments are important sources of methylmercury to lakes, then mercury in benthic invertebrates should be higher in lakes that are more influenced by their catchments. Now, METI was able to infer catchment influence by looking at more than 20 water chemistry and catchment variables. And what we can see here is that in lakes less influenced by their catchments, we're just giving a couple of examples here, compared to lakes more influenced by their catchments. So these are small lakes in big catchments with steep slopes. The percent methylmercury, this dark gray bar, doesn't change. However, what does change is the delivery of total mercury to the system. So A, it looks like methylmercury is being produced in a different environment than we originally thought. And B, it does look like total mercury is being delivered from these larger catchments and that they are really catchment driven. Our third prediction was that if catchments are sources of methylmercury to lakes, we should see methylmercury higher in the inflow waters than in the surface waters. Again, just two lakes, one with small inferred catchment influence, one with large. Gray is inflow, white is surface waters in the lakes. You can see here that percent methylmercury is like quite a bit higher in the inflow waters compared to the surface waters in Big Island Lake. In Sagway's Lake, it's actually extremely variable. So downstream of a slump that was dominated of mostly minerogenic material, not much methylmercury. Downstream of a beaver pond, which are known to be known methylmercury production sites, we see high percent methylmercury. And certainly we now know that methylmercury isn't just produced in the lakes, it's being produced in the catchments and is important in these lakes and is being exported to the downstream lakes. Now this is all opportunistic analysis. These are just little breadcrumbs that we're trying to, kind of pick up and then use to build a new understanding. And so where we're going with this is that we can't predict effects of change, whether it's climate change or forestry or any kind of resource development, without understanding how the ecosystem components are connected. In this case, the catchment and the water in the case of mercury. So results from METI's large modeling exercise indicated that we need to further refine our understanding, even though we're doing a pretty good job at the moment of explaining some of the fish mercury. And so we're going to look to be more systematic about this in the future. So METI's talk is tomorrow at 11:55 Saskatoon time in the aquatic ecology session. And I'd also like to give a shout out to human health and social science colleagues who have published on the link between fish mercury and human health, human consumption patterns, human concentrations of mercury and nutrient to mercury ratios. Thank you so much.
Thank you so much, Heidi. That was great. Sounds like a fascinating research topic, and it looks like there's some really cool fieldwork associated with that as well. So thank you very much for your presentation. Next up, we have Hadi Diaby. And so please feel free to share your screen if you have a presentation. You're just on mute.
Oh, there. Can you see the screen now?
Yeah, that looks great.
Okay. So we're switching over from watersheds to a little bit of a different topic over here with sewer sheds and how we're using wastewater-based epidemiology to track SARS-CoV-2 prevalence and variants in municipal sewer sheds. So this is done with the help from the funding from GWF, particularly the Environmental DNA Global Water Futures Program and many others. So how does SARS-CoV-2 enter our sewer system? So SARS-CoV-2 is released to the environment from an infected individual who may be asymptomatic or symptomatic. And this virus is then degraded in the sewer system where it releases the genetic material in the middle here. And that material is dispersed with the organic material in the sewer systems. And our goal is when we collect this, the sample at the treatment plants, how do we bring it back into the lab and analyze it to give us an estimate of how prevalent SARS-CoV-2 is in the community. So in spring of 2020, myself, Patrick Bredner, who's giving the next talk, will go into a little bit more of the detail. Naveta and Mark put on our masks, and we got onto the lab bench, and we started developing a method to measure SARS-CoV-2 in wastewater. Using what we knew from environmental DNA techniques of measuring genetic material in the in environmental samples, which are challenging matrix, as well as the wastewater treatment systems. So using the CDC guidelines in measuring of SARS-CoV-2 in environmental samples, we began developing a method to prepare the sample, including a matrix recovery control, which is a related SARS-CoV-2 coronavirus spiked into the sample, where we'll measure the recovery throughout the process of sample preparation. We then concentrate the genetic material using a PEG precipitation with centrifugation at a high speed. We then extract the genetic material, the RNA extraction using a kit from QIAGEN and the QIAcubes, where we measure the RNA signal and we test for inhibition, the human fecal control, the PEG-MMOV, which will be discussed in the next presentation, as well as a matrix recovery control and quantitative control. And we measure the same genetic signal that's measured in the clinical testing, the N1 and N2 targets in the SARS-CoV-2 genome. So in the summer of 2020, we began developing this method to see if we could apply it at wastewater treatment plants. So our two sites of that we began this pilot study were the region of Peel and the region of York, which covered 99% of the population in the region of Peel, and about 68% of the population in York region. These two sampling sites. In January of 2021, we began to sample at the three major treatment plants in the region of Waterloo, the Waterloo, Kitchener, and Galt treatment plants, which cover about 80% of the population in the region. So here's a schematic of the graph that I'll be presenting in the next slide. But here we can see that the clinical cases correlates pretty well with the wastewater signal in teal, and the clinical cases metrics that we used were cases by episode date that's measured, and the wastewater signal is just the averaging of the two genetic signals that the N1 and N2 in the SARS in the wastewater sample. And using this data in 2021, before the Omicron wave, where the clinical testing became overwhelmed, we see a high correlation between the two of 0.89. So here, this is the Clarkson graph again, and GE Booth shows that high correlation between the two signals, the teal being the wastewater signal and the orange being the clinical case data. And again, in the York region, we see this similar high correlation between the two wastewater signals and the clinical case count data. And the region of Waterloo has a similar correlation between the two sets of measures, but there's a lower magnitude in the wastewater signal as well as the case counts in the smaller regions. However, all sampling sites had a significant positive correlation that was significant. So this data is currently used by the public health units and the Ontario Science Table on public dashboards. And here we see the wastewater signal in the Peel and York regions, in the clinical cases being the blue shaded area, whereas the wastewater signal is shown here in the line graphs. And we see that there's divergence of clinical testing to wastewater signal that occurs in January of 2022, whereas when the clinical case counts became overwhelmed, clinical testing became overwhelmed with Omicron surge, and the province couldn't keep up with the clinical testing. So that's why I presented the data that showed the correlation between the two pre-Omicron in the previous slide. And here's a similar graph from the region of Waterloo that shows the wastewater signal in orange, as well as the clinical cases in purple. And this data is provided to a centralized data hub managed by the Ontario Ministry of Environment and Conservation Parks. And they provide this data to the science table, which projects a province-wide snapshot of what is happening in the wastewater in the entire province of Ontario. And here we can see that there seems to be the Omicron wave that began, and here's the BA2 wave. So using the wastewater sample that we collect, can we use this to track the prevalence of variants of concern? And here we see that yes, if we use qPCR specific assays to measure alpha and delta variants, we can see that it tracks really well. The wastewater signal tracks really well with the percent of alpha on the report date. So the bar graphs are the clinical testing, the percent clinical testing that have alpha, and we can see that there is an increase in alpha in late March, early April that peaks in mid-April, and then we can see the rise of the delta, and as well as our wastewater signal that shows that it links pretty well together. We also see that the wastewater followed clinical sequencing data during the Delta Omicron wave. And here we see the Delta dominance in late November in black, decreases significantly in mid-December. And the clinical cases here on the right show that the sequencing data from clinical were predominantly Delta, and then it switches to BA1 and then BA2 here in yellow. And so these VOC assays are currently being further developed to measure new variants such as the BA4 and BM4. And here's just a graph that shows how we're able to use this data to show the rise and fall of the different variants of concerns at one of the treatment plants over time. So here we have the alpha in blue, and then the switch over to the delta in orange, as well as the Omicron wave that began in mid-December, and then the BA2 specific assay that occurred in the early part of this year. And we continue to develop assays to measure specific variants of concern. So in conclusion, the wastewater-based epidemiology correlates well with clinical cases and can be used as an alternate to clinical case testing. As clinical case testing, it becomes overwhelmed with the surge in cases, and covers a wider demographic with less biases because everyone contributes to the signal. And wastewater-based epidemiology can determine variants of concern prevalence in almost real time as long as the qPCR assays are developed. And wastewater will continue to be important in the management of this and future pandemics. And with that, I would like to thank GWF for making this possible, as well as the regions for their support and insight into the project. And I will take any questions at the end of the presentation. Thank you.
Thank you so much for that, Hadi. That was fascinating and so incredibly relevant to what we are all going through right now in the current climate. Such an interesting and diverse group of projects so far. So this is just fascinating to learn about all the amazing work that you're doing. So up next, we have Patrick Bredner.
Thank you. I think we're wrong screen. Sorry. There we go. She's presenting fine now. So yes, I'll be building off of Hadi's presentation and looking more into some methodological questions regarding COVID-19 wastewater surveillance. So with wastewater-based epidemiology, as Hattie outlined, and I'll outline again briefly here, it has a central goal to inform public health of COVID-19 trends with the idea as more people become sick, you'll see an increase in the disease's genetic fingerprint in wastewater. We can see that in image 1 on the far left. To estimate the prevalence of COVID-19 in a community, we look at the SARS-CoV-2 virus and we use 2 gene targets, the N1 and N2 genes. And to account for some external factors such as fecal load, we can normalize our SARS-CoV-2 virus against an endogenous reference such as pepper mild mottled virus. Interestingly, pepper mild mottled virus is different in morphology, and it lacks the envelope that SARS-CoV-2 has, and it also is a rod shape, as where SARS-CoV-2 is spherical and has spike proteins. So expanding wastewater-based epidemiology, when we have a wastewater sample in step 3, we can bring that back into the lab and concentrate the virus. So in this case, SARS-CoV-2 and pepper mild mottled virus. We take the concentrate and we can extract the genetic material, the RNA, in step 5 and purify it and get it ready for final quantification in step 6 using quantitative PCR. The most important aspect of this is mobilizing that data that we generate back in lab to public health to inform of COVID-19 trends over time. My project focuses specifically, or the work I'll be presenting today, is focused specifically on the RNA concentration step. And what this is really asking is where the virus is between the solid and liquid fractions. I've opted to use a centrifugation-based approach to address this. So if we look at this conical tube in the left of this figure, the solid fraction would be the pellet after centrifugation, and the liquid fraction would be the supernatant after centrifugation. To analyze that supernatant further, we can process it through ultrafiltration, such as an Amicon unit on the far right. That essentially takes the volume of the supernatant and concentrates it down, passing it through a very small membrane. And you take that resulting concentrate and process that for RNA extraction and quantification later on. Additionally, as Hattie mentioned, for our routine analysis, we use polyethylene glycol precipitation, or PEG. This aims to capture both the liquid and solid fraction of the wastewater in at once, and you take the resulting pellet and use that for quantification. To do this, you require a high speed, upward of 12,000 Gs, and we use a 2-hour spin time for that. So today I'll be going over 2 pieces of data, and the first one is how does the partitioning compare between our analyte of interest, SARS-CoV-2 with the N1 and N2 genes, and against our normalizing endogenous reference PMMOV. And this was done at 12,000 Gs for 2 hours. We compare the solid and liquid fractions. And because we're just comparing the solid pellet to the liquid fraction that we use through the Amicon unit, you can get an estimation of the percent where this genetic material is found between the 2 fractions. Additionally, we will use our benchmark against the PEG precipitation. The second piece of data I'll show today is how does centrifugation impact viral recovery? And for this, we'll compare the what we use in lab, or 12,000 G for 2 hours, to a 4,000 G for 10 minutes. And the reason for the 4,000 G force and the 10 minutes is at the time of doing these experiments, it seemed like the prevalent setting used in literature to separate the solid and liquid fractions. So it was interesting to compare the common method against a more higher force and longer speed time. And again, we'll compare the solid and liquid fractions and PEG, our routine method. So looking here, this is on a log 10 scale, and it's the copies per tube. So that is how much of the liquid fraction with the supernatant, and what's the signal in the solid fraction, the pellet. Hence the percent between the solid and liquid fractions when no PEG has been added. And remember, when PEG has been added, it aims to capture both the liquid and solid fraction in one inclusive measure. We see N1 and N2 are 2 gene targets for SARS-CoV-2 track very similarly, and under this 12,000 G setting, split approximately evenly between the solid and liquid fractions, whereas PMOV, or endogenous reference, is much different in both quantity. It's much more abundant in wastewater, but it also partitions differently, where most of the signal is in the liquid fraction in the light blue. PEG precipitation across each of these targets helps improve our overall signal compared to just the solids alone. And that's because it's helping to bring down some of that liquid fraction through the precipitation. Next, if we look at how centrifugation impacts a recovery of each of these two viruses, for N1 and N2 is our SARS-CoV-2 targets. When we use that low force for a short amount of time, most of the signal is up in the liquid fraction. When we increase the force to 12,000 Gs for 2 hours, it helps bring it back down to that 50-50 split seen in that prior figure. And again, our PEG precipitation helps bring down some of that signal from the liquid fraction into our pellet and aids in our recovery of both fractions. For PMOV, we can again see that that is much different in both abundance, but also how it splits between the liquid and solid fractions. Interestingly, for PMOV, regardless of whether it was in the 4,000 G for a short amount of time or the 12,000 G for a longer amount of time, it remains in that liquid fraction and it's less influenced by the shift in centrifugation conditions. So conclusions of this is overall, our analyte of interest, SARS-CoV-2 for COVID-19 wastewater surveillance differs than our endogenous reference PMOV in both quantity and partitioning. In this figure, I just summarized that with SARS split between the the liquid and solid fractions, with pepper mild model virus mostly in the liquid fraction. And when we add PEG, it helps bring some of that signal down, but it's not as efficient, and that's denoted by the dashed arrow. And overall, this aids in the interpretability of the data and helps explain why so many different lab groups with such a diverse suite of methods are able to measure SARS-CoV-2 in wastewater. It's because if you look at the liquid fraction or the solid fraction or a combination of the both, because it splits between the two, you're able to measure it over time regardless of the approach you take. And more broadly, this supports the current method development for wastewater surveillance, but also for potential future infectious diseases. And finally, just like to thank you for your attention today and all the support I've had in this project.
Wonderful. Thanks so much, Patrick. And next up, we have Ana Cardenas.
Hi, let me see because I don't see where to share.
At the bottom, you should just see a share screen button.
Oh yeah, it was hide. Can you see my screen now?
Yeah, looks great.
Okay. Hi, my name is Ana Cardenas, and I'm a student in University of Saskatchewan. And this presentation is about impacts of wastewater effluents and seasonal trends of emerging contaminant in water and sediments of two cold region rivers. So lately it have been more and more common to find news like this. Products turn guppies into zombies. This is about a study conducted in Australia, or this one, fish on volume. And this is a study conducted about 58 different pharmaceuticals and in a single fish. So what are the causes, the effects, and what we can do about it? Rivers are a fundamental part of ecosystems as a source of fresh water and habitat for aquatic organisms. So river quality has been improved significantly in many developed countries due to the widespread installation of wastewater treatment plants. Most of the emerging contaminants are found in minimal concentrations in the receiving water bodies, but still due to their continued release and high biological transfer potential and possible bioaccumulation in aquatic organisms, they may be an important factor in the ecotoxicological risk of effluents. Wastewater treatment plants are not specifically designed for removing emerging contaminants, and these compounds practically remain in the treated water. The concentration of pharmaceuticals in environmental media can be affected by several factors, apart from the environmental ones, including temporal variations, the flow rate of water receptors, the sorption capacity of sediments, and other abiotic dynamics processes that operate in aquatic ecosystems. Based on the variations of emerging contaminants, the main objective of this research was to investigate their changes over the time and the relationship with physiochemical parameters present in 2 wastewater treatment plants located in Saskatchewan, Canada, as well as the risks associated with the ionizable organic compounds, specifically pharmaceuticals. So these are the location of this study. So the South Saskatchewan River is one of the largest and most important rivers in Saskatchewan. It begins in Rocky Mountains in Alberta, and Wascana Creek is an effluent dominated stream nearby the city of Regina, constantly receive effluent from the Regina wastewater treatment plants, and is part of the Coapella River basin that is located in the southern part of the province of Saskatchewan as well. Some stressors of water quality in the South Saskatchewan River and Wascana Creek include contaminants entering the water body. Downstream water quality in both water bodies can be affected by the constant discharge of effluents. Considering all these conditions that can offer reliable information about water quality, the samples were collected in South Saskatchewan River and Wascana Creek, upstream and downstream the wastewater treatment plants. So initially, I prepared the samples for the analysis, and then a solid phase extraction was conducted for both, for water and sediments. For the diffusive gradient in films, the DGT as standard configuration was used, and all the gels were prepared with the 1.5% of agarose, and the binding gels were extracted using methanol. For the identification and quantification of the target pharmaceuticals, I used the LCMS using a positive ionization mode, and the QSAT orbital method was implemented for all the data acquisition, and the processing was conducted using Excalibur software. For the analysis of the data, because the concentration of the compounds obtained from the conventional samples of water and sediments was firstly descriptively, then I needed to use a multi-level model for comparison of all the variations. The results obtained were analyzed considering the responses in water, sediments, and physiochemical parameters in the developed model. The different levels confirmed by the time of pharmaceutical compound, the site, and the time were compared between the sampling places through the pairwise comparison using a t-test with the Pulis standard deviation, and I used bone Ferroni correction for the analysis that were performed all using R for visualization and processing of the data. In this figure, you can see all the results for the physiochemical parameters. The water temperature results were similar upstream and downstream for both sides. pH values were higher in South Saskatchewan River than Wascana Creek. Upstream the South Saskatchewan River, the values were lower than downstream, and the opposite trend were found in Wascana Creek. The dissolved oxygen values in the South Saskatchewan River were constant within the 3 seasons. In Wascana Creek, the values were changing depending on the season. About the specific conductance, the values were higher in Wascana Creek, whilst in Wascana, always increasing after the wastewater treatment plant for both sides, for South Saskatchewan River and Wascana Creek. In this plot, we can see the concentrations, the logarithm of the concentration to facilitate the comparison. So significant variations were found between the compounds, the pharmaceuticals, and this led me to use the multi-level model I mentioned before, individually per pharmaceutical. So for instance, for carbamazepine and lamotrigine, the concentrations in water show variability related to the site, the time, and the location with respect to the wastewater treatment plant. For amitriptyline and bupropion, only time and site. For clozapine, time and location. And for venlafaxine, only site and location were significant. Overall, pharmaceutical concentrations in water and sediment samples were higher in Wascana Creek, and similar trends were obtained using the DGT samples. After comparing the results of the water samples analyzed by conventional GRAB and DGT, carbamazepine was only found using the puzzle sample. Sediment samples indicated significance in the variation of the concentration of pharmaceuticals related to the location, the temperature, and the dissolved oxygen. In the next slide, you are going to see tables, and these tables are a comparison with information from previous research. So this first one is for conventional GRAB of water. So from my study, we could see that the highest concentration in water was amitriptyline. At the lowest one was fluoxetine. Only fluoxetine concentration were higher in South Saskatchewan River. The concentration found in the water samples, excluding fluoxetine, were all above the ones presented by previous study. And amitriptyline and bupropion were above the critical level presented by previous report. This table is a comparison with the water samples obtained using the DGT. And with the DGT, we found a coefficient of variation below 30% for 97% of the samples. And this indicates the homogeneity of the data in comparison with the conventional graph. Passive samples are relatively new techniques for water sampling. And this is why you can see the comparison with different techniques here. Overall, the concentration of pharmaceuticals obtained in this study, again, were higher than those presented by previous research. 4 out of 7 pharmaceuticals analyzed have concentration above the range indicated by similar projects. In this table, we have the comparison for sediments. Sediments have higher concentration of pharmaceuticals than water for 95% of the samples. Amitriptyline was the most abundant pharmaceutical, not only in water, but also in sediment. The highest concentration in sediments were for clozapine, and the lowest concentration was for venlafaxine. Pharmaceutical concentration were higher downstream than upstream for both sides. So in general, to our best knowledge, this research was the first to comprehensively analyze and investigate the occurrence and phase of pharmaceuticals in wastewater treatment plants in Saskatchewan. The data obtained on the distribution of pharmaceuticals in the South Saskatchewan River and Wascana Creek is indicating the exposure of aquatic organisms to wastewater treatment plant effluents, which are mixture of pharmaceuticals of concentration between nanogram and microgram per liter. Water samples by conventional graph using in comparison with the DGT results less sensitive, and the homogeneity in the DGT results were higher, and this is leading to get small concentrations easily using the DGT. Amitriptyline was the pharmaceutical with the higher concentration out of 7 pharmaceuticals evaluated in water and sediment samples at the 4 sampling sites and across the 3 seasons. Concentration of pharmaceuticals were several orders of magnitude higher than the concentration obtained by similar studies. And these findings are significant since both the South Saskatchewan River and Wascana Creek are important drinking water reservoirs and is in the habitat for fish species that can be affected for those concentrations of pharmaceuticals. While this study was focused only in 2 regional water bodies, these findings are useful in evaluating the risk of wastewater treatment plants discharged into water bodies in other semi-arid regions globally. So thanks to my founders and the people that have been working with me in this project. And if you have any question, please let me know. Thanks for your attention.
Thank you so much, Anna. That was fantastic. I noticed a couple of people joined us throughout Anna's presentation. So just so everybody's aware, we're holding off questions until the very end. We are really great on time, so we should have anywhere from 10 to 15 minutes at the end of the lightning round to have some discussion and some questions. So Anna, if you'd like to just stop sharing your screen, we can pass it on to Blake, who's going to begin the 2-minute lightning talks. And for those who are doing those talks as well, you will be sharing your posters on your own screen, just so you're aware.
Can you see and hear me okay?
Yep, looks and sounds great, Blake. Thanks.
Okay, thank you. Okay, so hello everyone. My name is Blake. I'm a master's student at the University of Waterloo. And today I'm going to be briefly discussing how wastewater can be used to inform targeted public health measures using a rapid and affordable passive sampling approach. So the first thing that you need to know, and probably already know from the other talks, is that individuals who are infected with COVID-19 shed gene fragments of the virus into wastewater. With more than 89% of Canadians relying on municipal wastewater infrastructure every day, detecting these fragments in wastewater wastewater can be a reliable and independent way to estimate infection prevalence in our communities. So here we've used a passive sampling approach to monitor 3 university residence complexes over a 9-month period. Essentially, a piece of gauze was submerged in the sewer channel at a site downstream of all 3 complexes here. And using RT-qPCR, we can detect the presence or absence of these gene fragments to identify where an infected person or persons may reside. In the event of a positive signal at the downstream site, we would move to the 2 upstream sites the next sampling day to identify the source. This was successful in fall 2021 when case counts were comparatively low. We were actually able to identify 1 individual with the virus who was infected 1 day before they became symptomatic and 4 days before clinical confirmation by a PCR test. So this was 1 individual in a population of over 1,000 students, which is pretty remarkable. In winter 2022, however, the Omicron variant produced a major spike in cases globally and infection was widespread. And at that point, our objectives shifted to only monitoring the downstream sites for declines. So if you look at this graph on the right here, you can see the 2 major Omicron peaks we observed on campus, which really dwarf that single signal that we observed in the fall from that 1 known case. So while passive sampling is a semi-quantitative science, it has proven useful in acting as an early warning system for potential virus outbreaks. And this principle can be applied to other infectious diseases and congregate living settings where vulnerable populations reside. Wastewater-based epidemiology has been placed at the forefront of the fight against the COVID-19 pandemic and will certainly be a critical component of pandemic preparedness in the future. Thank you so much, and I welcome any questions or comments that you might have on my poster. Thank you.
Amazing. Thank you so much, Blake. That was fascinating and incredible that you guys were able to locate that single infected person. Okay, great. So we will go next to Sergey Bokenyov. Sergey, I'm not sure if you're speaking right now, but I think there might be something.
Okay, can you?
Yeah, there we go. Yeah, we can
okay.
see and hear you now.
Okay, our poster is about sensitivity of lake hypoxia to atmospheric physical forcing, such as air temperature and wind speed. And one of the first studies to explore the differences between the simulated first order and the second order effects. I would like to remind that first order effects are those caused by changing only 1 parameter at a time, while the second order effects are those caused by simultaneous changes at a time. So what is lake hypoxia? It is the bottom water dissolved oxygen concentrations below 2 milligrams per liter as the result of excessive nutrient loads and elevated algal biomass. Apart from nutrients and algal biomass, it is largely controlled by water column thermal stratification, which is a function of air temperature and wind speed. Lake Erie. Lake Erie's central basin and its hypoxic conditions are extremely sensitive to atmospheric forcing and future climate change because of the shallowness of that bottom layer of water. So now I would like to show how sensitivity studies to atmospheric forcings are usually made. Here you can see a number of scenarios of changing air temperature and wind speed. For example, here scenarios changing air temperature by, for example, decreasing air temperature by 1 degree or increasing. And here you can see changes done to the wind speed. Here you can see 100% of wind speed and 0% of air temperature. You can see the base case scenarios. It could be certain here. And then what is done, so the people usually, they do sensitivity studies by changing air temperature, like keeping the wind the same, You can see this red square scenarios, or they can, for example, keep the air temperature the same, but changing the wind speed. For example, you can see those scenarios. And from those scenarios, they can some kind of project what would be the future climate with different wind speed and air temperatures. So all those scenarios with a combined effects, so they are second order effects. Second, but they're not usually not done, those highlighted by the blue color. They're not usually done because it's like too much for simulation work. But so it's assumed that the second order effects are assumed that the sum of corresponding first order effects. For example, effects of A for air temperature, or W for wind speed, X and Y superscripts that denote for the modified changes to air temperature and wind speed. So in our work, we decided to see, for example, if those how they differ, the effects from the combined cases are different or not from the effects of the sum of the corresponding effects. So what we did, we applied a three-dimensional lake model to estimate the effects of different meteorological forcing on dissolved oxygen. So we applied a very complex coupled hydrodynamic and water quality model. And here you can see only like a simplified dissolved oxygen cycle or sub component of the model, and some kind of the processes that we included in our work, like major and the most essential processes. After like running a number of the scenarios, we plotted the results from second order effects against the sum of corresponding of the first order effects, and used the linear regression model to understand the relationship between them, whether they form the same relationship or they come from the different populations, so they differ. And for the results, I would like to ask you consult our poster, or when we have all those results presented, and they're quite interesting. So, and this is like, I want to highlight this work is like first in its origin, because it has never been before, like comparison of the first order effects with the second order effects. So I'm done with my presentation. Yeah.
Perfect. Thank you so much, Sergey. Looking forward to seeing the final poster, and great to hear you're doing such new work. So if you'd want to stop sharing screen, Sergey, whenever you can, we'll pass. Awesome. Thank you. We can pass it on to Eric Frichette.
Just one moment.
And Sergey, please feel free to put your contact info in the chat as well whenever you get a chance, and people can reach out to you for that final poster.
Okay, can everyone see my screen okay?
Yep, looks great.
Beauty. Okay. Hi, my name is Eric. I'm going to be talking about my poster, which involves the development of a particular zooionic polymer antifouling coating for dissolved oxygen surface water monitoring. So Indigenous communities across Canada are very disproportionately affected by poor water security, and that's due to a wide variety of historical, political, environmental factors. And because of this, knowing what's in the local community surface water can be very beneficial in knowing how to improve water security in a number of ways. The Mackenzie-Boston Creek subwatershed is the major surface water source on 6 Nations of the Grand River, apart from the Grand River itself, which is right next door. And this covers about 3/4 of the community's surface area. However, there has been little characterization of this watershed and absolutely no live monitoring characterization of this watershed. But any inexpensive sensor-based live water quality monitoring that would get put in a sort of environment would immediately yield very fouled sensors. And now this causes inaccurate data to be acquired and also requires effort to re-clean these sensors.
Sorry, Eric, I don't want to interrupt. We're just seeing your presentation screen, so we can see your notes. And your slides are just a little bit small up at the top. Perfect. There you go.
Okay, sounds good. There we go. So to help combat this problem, we've been in development of a series of water quality monitoring stations that at this moment monitor for 5 common parameters. The dissolved oxygen probe is one of these sensors and works by oxygen diffusing through a dense PTFE or Teflon membrane. By coating the membrane in a series of zwitterionic polymers, we can see reduced fouling on these coated membranes due to a pronounced hydration layer. So that's these water molecules are on top of the zwitterionized PGS copolymer. They create a physical barrier between the zwitterionic copolymer and any potential foulants like proteins or bacteria. And so we want to know whether the DO meter data accuracy will be affected by the zwitterionic coatings, because it's not going to matter too much in reducing anti-fouling if your solution to the fouling problem prohibits oxygen transfer just as much. So first, the anti-fouling properties of this SBMA copolymer were improved by altering zwitterion graft time and the reaction time of the intermediate adhesion layer. Then DO diffusion experiments were performed for different fouling conditions and membrane thicknesses. And one of the results shown here on the bottom right, where a 10 micron thick PTFE membrane was subjected to environmental fouling for 2 weeks. And you can see a substantial difference in the response time in the final steady state concentration when diffusion experiments use the unfouled membrane versus the fouled membrane. And currently we're working on seeing how the zwitterionic copolymer itself affects oxygen diffusion levels in this setup. But overall, these polymers look like promising material for anti-fouling use on our DO sensors in the field. And that's all I've got there.
Great. Thank you so much, Eric. That was fascinating. Perfect. So yeah, we're on a really good time so far. We have 4 more minutes until the hour. So we will finish off with Sarah Packle-McCormick. And then we have 15 minutes for questions and discussion.
All right, so hopefully everyone can see my slides. Good. All right, so thanks everyone. I just wanted to talk a little bit about our poster, and hopefully that will entice you to go see it, the full version. And so traditional foods are very important for Indigenous communities in Northern Canada, and this includes locally caught fish. However, there's a gap in the literature about mercury bioaccessibility in freshwater fish species from Northern Canada. So we were able to get some fish samples from part of Dr. Heidi Swanson's ongoing work in the Northwest Territories for 4 different freshwater fish species. And what we did is we chopped them up and we then digested them in a model of human digestion in some simulated gastric juice and some simulated intestinal juice. And then we, after our digestion and shaking at body temperature, we filtered out any undigested chunks of fish, and we measured the mercury concentrations in that fraction at the end. So in that gastrointestinal juice. So a little bit about our results. We looked at the percent mercury bioaccessibility. So the percent of the mercury from the original concentrations in the fish tissue that were then present in the gastrointestinal juice that we simulated. And we found that the percent mercury bioaccessibility differed based on fish species and also based on the lake that they were from. Additionally, for some of the larger fish samples, we actually cooked some of them. We pan fried them in water. And we found that mercury concentrations actually increased a bit in the cooked samples. And we think that's because of moisture loss due to steam that was made during the cooking process. But we found that the percent mercury bioaccessibility was actually a lot lower. So overall, the concentrations of mercury that were in those gastrointestinal fluids, they were lower in the cooked samples compared to the raw samples. So as you can see here, that was the case for lake trout, lake whitefish, northern pike, and walleye. The cooked samples, their bioaccessible concentrations were lower in the light blue compared to the raw samples in the teal color. So hopefully that's a little bit of information to pique your interest, and you'll check out our full poster. Thank you.
Wonderful. Thank you so much, Sarah. That is very interesting and definitely makes me wish I could come check out the full poster tomorrow. Okay, great. So we're just about a minute away from the hour. So we have 15 minutes of question and discussion time, which is fantastic. So if anybody wants to kick off with some questions, please feel free. I do have one for Blake if nobody else wants to speak up first. Blake, I'm super interested to see how you guys were able to identify the single infected person from, for lack of a better word, a downstream sample, how you were able to trace that back to that single person.
Yeah, absolutely. So like I said, we had a tiered sampling approach. So you'd have the downstream site that you would monitor regularly. And so once we saw this massive signal that we'd never seen before, the next sampling day, we moved upstream. And so by measuring all of those 3 sites, we can likely exclude the possibility of that signal coming from elsewhere when the upstream sites are negative. And so working closely with our campus partners in the housing department, we were able to attribute it to at least one known case. And then we have the dates for the testing and the dates for symptom onset. And so we were able to conclude that that signal came from at least one infected individual.
Wow, very interesting. And were students informed of your study, and did they kind of provide any of their own information to contribute?
Yeah, so they were aware of the project, and the housing department actually had set up a dashboard to communicate with students when their residence was positive or a positive signal was detected. But all of the information that they provided in terms of their own personal health information, like PCR test results, that was all provided to the housing department. So we were able to work with them to clean that data up and work with it. So that's next on my list, seeing how those known cases correlate with the signal over time.
Yeah, wow, awesome. Very, very cool. And I think a residence or a university dorm is just the perfect sample site to do that work. So very cool.
Absolutely, yeah, thank you.
Awesome. Anybody else have any questions, comments, just discussion points they wanted to mention? Okay, perfect. Well, if any of the presenters haven't put their contact information in the chat, or if anybody wants somebody's contact information specifically who hasn't added it, please feel free to add anything into the chat box. I'll leave things open for another minute or so, just in case there's any last minute questions or comments. And then we can all sign off and have a little break before the next session or whatever everybody has up next. Thank you all so much for attending. It's been really wonderful to learn about your very, very interesting and diverse areas of study, and definitely interested in keeping in touch with a couple of you to see how things go further with your work. I guess I'll add my info in here too, if anybody has. Perfect. Thanks, Steph. Thanks, everybody. Awesome. Well, it doesn't look like there are any other questions or comments. Again, great to meet you all and listen to your work. And I will, sign off and hope everybody has a wonderful afternoon.